Very good. Welcome to Digging In. My name is Anya Crowley, and I'm hosting for Lindsay Randall today as we welcome participants to enter our Zoom webinar. We will get started in just a moment and be able to introduce our guest speaker today. So please come in, settle down with a nice beverage or lunch, and we'll get started in just another moment. Okay, very good. I'll get us started with a few introductions. Once again, my name is Anya Crowley. I am a trustee of the Massachusetts Archaeological Society. I am filling in hosting duties today for our regular host of Diggin' In, Lindsay Randall, who has another obligation this afternoon. But on behalf of Mass Archaeology and the Robert S. Peabody Institute of Archaeology, our co-host, I want to welcome everybody who's joining us today. Diggin' In is a series of live presentations with scholars from around the country, co-sponsored by the Peabody Institute and Mass Archaeology. We're going to begin with a land acknowledgement for the land that the Peabody Institute itself and the School of Phillips Andover, Phillips Academy is on. So let me just read this in its exact wording. Phillips Academy occupies the land of the Pennacook and the Patuxet peoples and the lands of the contemporary Abenaki, Massachusetts, Wampanoag, Wabanaki, Pokanoket, and Nipmuc nations. We honor all Indigenous peoples who are here now, have been here for time immemorial, and will be here in the future. We acknowledge Indigenous genocide and the continued repression of Native peoples, voices, cultures, and spiritualities. We understand how education has been used by settler institutions in the attempted erasure of Indigenous peoples. We commit to interrogating the histories of and our complicity in colonization, centering Native voices and communities, and dismantling the ongoing legacies of settler colonialism at Phillips Academy and beyond. Just a little plug for our Diggin' In program. Join us every other Wednesday at 1:30 p.m. through December for our presentations. For a schedule of dates and presenters, please visit us at peabody.andover.edu or at the Massachusetts Archaeological Society's Facebook page at Mass Archaeology or our website, massarchaeology.org. If you are enjoying our programming, and we certainly hope that you are, consider expanding your impact by becoming a member of Mass Archaeology. We're able to bring projects and programming like this through the support of viewers and members like you. So enough of our preamble. Today, I want to welcome our speaker, Anya Gruber. Anya Gruber is a PhD candidate in anthropology at the University of Texas at Austin. Her research is focused on paleoethnobotany in New England and public outreach. She's also a writer and a science communicator. That's something close to my heart, so very glad to have you with us. And just by way of housekeeping, at the conclusion of the talk, viewers will be able to submit questions directly through the Q&A function on the bottom of your Zoom screens. Please feel free to do so. We will then give our speaker a few minutes to answer a couple of those questions, as many as we can, with the understanding that we may not be able to get to all. So without further ado, welcome again to our guests, and welcome Anya, and thank you for being with us today. The floor is yours.
Thanks, Joanna. Okay, I'm going to share my screen. Okay, so thank you so much for that introduction. So today I'm going to be telling you all about phytoliths and some of my dissertation work in Plymouth, Massachusetts. So as Joanna said, I am a paleoethnobotanist and environmental archaeologist, a PhD candidate. I am also a graduate of the UMass Boston Historical Archaeology Master's Program. And I was recently a public outreach intern at the Northeast Archaeological Resources Center program. But my dissertation research is focused on paleoethnobotany in colonial Massachusetts. So Joanna just gave a land acknowledgement for the Peabody Museum, but I just want to give another land acknowledgement. And although these kinds of territorial acknowledgements are a very small gesture, I do feel it necessary to take a moment to recognize the ongoing work to disrupt over 400 years of Indigenous erasure, and also just to show my gratitude to the Indigenous people of Massachusetts because my work is indebted to them. So our story today takes place in Patuxet, otherwise known as Plymouth, and in the 1600s, and our protagonist is the humble Phytolith. So some of you may have heard of the Phytolith, some of you may not have before, but my goal today is to tell you all about them and how important they are and how interesting they are. And I hope that by the end of this, you will love Phytoliths just as much as I do. Okay, so a little bit of background on Plymouth/Patuxet. This area is part of the original homelands of the Wampanoag people, who are just one group of Indigenous people in New England. And the work that I do is in collaboration with the Plymouth Colony Archaeological Survey, which was started in 2014, and I became a part of it in 2015. And kind of stuck through all the way through 2021, which was, we went out in the field last year, with of course a break there for 2020. But this project is an ongoing work that is part of Plymouth 400, which has been a effort by the town of Plymouth to commemorate the 400th anniversary of the arrival of the passengers of the Mayflower, otherwise known as the Pilgrims. And it is headed by Dr. David Landon and Dr. Krista Bronick of UMass Boston. So with this project, one of the main goals of this research is to identify and describe and analyze some of the Indigenous sites that are in Patuxet, in Plymouth, as well as understand the earliest years of English colonialism, kind of starting in 1620, and try and identify some of the oldest household contexts from this area. And one of the research questions that I have been most interested and most involved in is what were the ecological consequences of colonialism from this time? And how do we continue to see those today? And how do they impact people in the 17th century? So what the heck is a phytolith? So some of you may be asking this. The word phytolith comes from the Greek roots for plant rock. So that kind of encapsulates the essence of a phytolith. And they are microscopic silicified plant cells that are left behind in the dirt when a plant dies. So basically the way that these are formed is that when plants grow, they absorb nutrients and they absorb water through the soil. And when they do that, they also absorb dissolved minerals that are in the ground or dissolved in the water. And when they do that, this silica-based material, these molecules, then become incorporated within the plant tissues themselves. And they kind of settle between, in the spaces between plant cells, kind of around the cells in the tissue of a plant, whether that's a leaf or a stem or the flower. And they make these kinds of imprints or a cast of the plant cell, and they kind of take the form of whatever that cell looks like. So since these phytoliths, these silicified plant cells, are made out of inorganic materials, they're made out of silica-based compounds, they do not degrade or decompose the same way that the living tissue of a plant does. So a living tissue of the plant is carbon-based, it is organic. So that is going to kind of go through the processes of decomposition. That is, unless it is in some kind of ideal environment. So if it's frozen or if it's very arid, then those processes might not kind of happen that same way. But somewhere like New England, where we have a pretty harsh environment, we go through freezing and thawing cycles. That's very hard on organic materials. But these inorganic phytoliths remain because they don't degrade that same way. Okay. Like I mentioned, that these phytoliths are cell sized, so they are microscopic. You cannot see them without a microscope. To kind of give you a kind of perspective on how small they are, an individual phytolith is about the same size or smaller than the width of a human hair. So they are very little. Okay, so how do you find phytoliths? If they are microscopic, then how do we find them and how do we look at them? So a lot of these principles are similar to pollen analysis, which is a little bit more well-known, and you may be a little bit more familiar with the processes of pollen analysis. So some of this might sound familiar, but phytoliths are found in archaeological sediments. So wherever there's a context, whether that is in an open excavation unit or if you're taking a soil core, any kind of archaeological significant context may be useful in terms of phytoliths. So I'll get in a little bit more about what kind of context phytoliths are most useful in, but there is a wide variety of types of excavations in which this might be relevant. So the biggest thing with collecting sediment, whether it's pollen or phytoliths, is that they need to be collected in a sterile bag and minimize cross-contamination as much as possible. They want to be as clean of a sample as possible. And then it's off to the laboratory. So when I take soil samples into the lab, what I then do with them is go through a process of chemical rinses and these different kind of chemistry processes where my end goal is to separate the actual phytoliths from the rest of everything around it. So I rinse the samples in hydrochloric acid, and that dissolves any little bits of calcium carbonate. So shells or bone, anything that I don't want to be in my sample, anything that's not a phytolith. And then I separate it out from any clay particles because clay particles are heavier than the phytoliths. So I put it in some water, the phytoliths float, and everything else we get rid of. So once I have separated the phytoliths from all the surrounding dirt, I put it on a slide and I do a permanent mount on the slide. So I put a cover slip on it and then I keep it in my little boxes, which I have behind me, and I look at it under my microscope, which I also have behind me. This is my little work from home setup. This is my microscope. Her name is Rosalind. So Rosalind helps me look at these phytoliths and I identify them and I count them in a long, sometimes painstaking process, but that is what I do. And then I use those phytoliths to answer my research questions. So when I say identify a phytolith, this is also a similar process to pollen analysis, where phytoliths take different forms. They have different shapes, they have different sizes, they have different characteristics. You can see here on the slide, this is just an image with lots of different types of phytoliths. And you can see that some of them are kind of rectangular and sort of kind of chunky. And some of them have like these kind of dumbbell sort of shapes. Some of them kind of look like a cartoon of a bone. Some of them are long and skinny and kind of spiky. So you get all kinds of different looking phytoliths. I kind of describe them as being a little bit like snowflakes. They all have these unique shapes. So based on those shapes, I can use the metrics and the categories that have been established by scientists over the years. And I can use those shapes and these different sizes and correlate these phytoliths that I see to different kinds of plants or to a part of a plant, which can then be a proxy for me to be able to figure out what kind of plants were present at a site. So grasses, we are just lucky in that grasses tend to produce many phytoliths. A grass plant will produce just lots of individual phytoliths, and they also produce very varied phytoliths. So they can be really useful. And grasses and the grass family is really interesting is really helpful if you're interested in diet like I am, because some important grass species are maize and wheat and reeds and other useful plants to people. So like I mentioned before, lots of different archaeological contexts can be kind of relevant for phytolith analysis. So if you want to figure out what people were eating, then something like a field or a garden or a cellar or a kitchen could be really useful, or a hearth. So a lot of these kind of contexts that are helpful for sort of paleoethnobotany in general are also really helpful for phytoliths. And the other thing that's really interesting about phytoliths is, unlike pollen, which falls and disperses in pollen rain, phytoliths tend to stay where they are. So this can be a little bit complicated, of course, with different kind of formation processes and ways sites are formed, whether that's kind of natural changes or from people building or all kinds of different, we know as archaeologists, there are many ways that a site could be impacted. But kind of writ large, a phytolith is likely to stay wherever the plant that produced it was growing. So it's really good for that kind of hyperlocal analysis. So here are just some photos of some different phytoliths. This is what it looks like when you are looking through a microscope at a phytolith slide. And you can see there's a big variety in what they look like. And I've circled the actual phytoliths, and you can see that it's sort of surrounded by all sorts of other stuff. It's just kind of little bits of silica things that weren't removed during extraction, or just little bits of plant crap, like who knows what's under there. So one of the challenges is to actually figure out what is phytolith and what is just random stuff. So you can see that's what they look like when I'm actually identifying them under a microscope. Okay, so what can phytoliths actually tell us? So phytoliths are, sorry, I just lost my notes. Okay, so phytoliths I see as sort of one way in our toolbox of how to tell a story about the past. So phytoliths can help us understand plant and human relationships. They can tell us about diet. They can tell us about how people have managed their environment, how they have changed their environment. They can tell us about, like I mentioned, what kind of crops people grow, what kind of crops people grew, what were people growing in their gardens, what were people growing in their fields? Is that different? Is that important? If you are taking samples from a cellar, maybe is that where people were storing food? What kinds of foods were they storing? What were they kind of growing for sustenance over the winter? Excuse me. You can also focus on certain types of environments. So like salt marshes are very important for coastal Massachusetts. We have a lot of salt marshes. So how did those change over time? Those are usually pretty important environments for people. What were people kind of doing to or in these salt marshes? What kinds of grasses did people use to build their homes? What did they use to make baskets or mats or other items that were used in daily life or that were used to trade for economic purposes? And maybe how were people kind of on a large scale changing their environment? How are they coexisting with their environment? Did they practice cyclical burning? Did they clear forests for fields? There are so many different directions that we could go in with phytoliths. Okay, so hopefully I've convinced you the phytoliths are really cool and really pretty and really fun to look at. But I can imagine that many of you are anthropologists, and I am getting my PhD in anthropology. So where the heck's the anthropology? So this series has been, the speaker series, Diggin In, has been highlighting underrepresented, historically marginalized voices, historically marginalized histories. So I kind of took that as a good starting point. How can phytoliths support principles of decolonization? So a pretty big question, but I think it's a really important and interesting one. So phytolith analysis is, it draws from chemistry, it draws from physics, draws from math, it draws from biology. So it is a qualitative science. It is a quote unquote hard science, but I don't think that means that it should be disconnected from anthropological theory from human stories and from human experiences. So I see phytoliths as a really useful way of providing physical evidence that can work alongside and in dialogue with so many different theoretical points of view. It can provide this kind of qualitative side to human stories. So I'm just going to pull out two examples, two kinds of schools of thought that I am particularly interested in and draw from a lot in my dissertation work. And those two are political ecology and traditional ecological knowledge. Okay, so political ecology, you may be familiar with this, is essentially the way that we think about how humans and the environment interact, especially in terms of political and socioeconomic structures. So as Michael Watts put it, it's seeking to understand the complex relations between nature and societies. Now, those are two very big things. But in archaeology, and perhaps in paleoethnobotany specifically, it refers to the way that physical landscapes and physical environments intersect with social and economic structures. And this is particularly relevant and important in colonial contexts, such as in Plymouth, where these colonial regimes may be coming in and interacting with and sometimes dominating or having these complex interactions with Indigenous ways of seeing the world. So I see phytoliths as potentially acknowledging Indigenous agency in these kinds of colonial environments. And I also see it as particularly relevant to work against these kinds of colonialist assumptions of this pre-colonial pristine landscape, which was kind of the way of thinking for a very long time, that Indigenous people made no marks on the landscape. And that's kind of a pervasive thought even today, but that's just not true. And phytoliths and phytolith analysis could be a really interesting and important way of kind of talking about that. Okay, and then so traditional ecological knowledge, as Robin Wall Kimmerer defined it, is a knowledge, practice, and belief concerning the relationship of living beings to one another and the physical environment. So kind of some intersections with political ecology as well. And in the Americas, traditional ecological knowledge typically refers to Indigenous knowledge. That's not necessarily the only traditional ecological knowledge. That is generally how I think about it, and often how it is in the Americas. So thinking about traditional ecological knowledge, how can we use kind of place-based situated knowledges and oral histories to inform the way that we interpret phytolith data, and how can it frame our analysis and the way that we actually think about the results of these kinds of qualitative data collection. So I already used this example. I'm going to bring it up again because I think it's a great example, and that is the Indigenous use of fire. So in Massachusetts, cyclical burning was a very important and longstanding way of managing the environment. It was used to clear underbrush. It was used to enrich the soil, made it much better for agriculture, made corn grow much nicer. It made these meadow-like landscape that attracted deer, which are another important food source. So cyclical burning is absolutely practiced in this area, and also in among many different communities across the Americas in general. Is very common practice. So how can I use kind of these oral histories, these traditions of fire? How can I listen to these kinds of traditions and have that help inform the way that I interpret my phytoliths? Can I see these evidence of meadows with grass-dominated species that may thrive in the years after a fire? Is that present in the record? If I didn't know about fire traditions and cyclical burnings, would I pick that out in the phytolith record? So this is another way of, as I see it, going about understanding these data. Okay, so perhaps the moment you've been waiting for. I'll talk a little bit about phytoliths and some of my work specifically in Plymouth and Patuxet. I'll talk a little bit about some of my preliminary findings. So I'm still in the relatively early stages. I've done a lot of data collection, kind of working through the interpretation and what it all means, which is of course the hardest part, but we are getting through it. So these are just some broad patterns that I have seen already in the data. So one of my questions going in, and kind of one of the questions that we've had working in some of these early domestic colonial households, domestic contexts, is what kind of plants were important to colonists in the earliest years of the Plymouth colony. So we're talking 1620s. So building houses was very necessary. It was very cold in the wintertime. They needed shelter. And the colonists used natural materials to build houses. So they were somewhat ephemeral structures, and they were made out of grasses, and they were made out of dirt and mud. They were made out of sand. So these are referred to as wattle and daub houses. So what I wanted to know doing the phytolith analysis was, when I took samples from some of the daub that we found in excavations, was, can I see what kind of plants these colonists were collecting to make their roofs and to use as wattle and some of the plant material used to reinforce the walls of the houses. So I have found in some of the daub samples a lot of what are called flabellate bulliform phytoliths. So you can see that here on the bottom of the screen. It is not my microscope photo. I didn't have a good photo of these, so I used one from a publication. But that is what reed phytoliths look like. So reeds, reed grasses are very common in salt marshes. If you drive by the coast, you'll see these tall grasses with kind of like feathery tops that are kind of blowing in the wind. And those are Phragmites, which is common reed. And I found a lot of phytoliths from this plant in these daub samples. So I am interpreting that as probably these marsh-loving grasses were used in house construction. And another note that I thought was kind of interesting was, in these daub samples, I also found innumerable sponge spicules, which they are basically, they look like a phytolith. So these are remains of actual sea sponges. And they kind of just look like hollow spikes. And I don't count them because they don't really tell me anything besides the fact that they are there and they are present. But that is a really good indication that they were also using beach sand in their daub to kind of reinforce it, which also makes sense. But it was really interesting to kind of see that confirmed, that all these sponge remains were in this daub. So yeah, beach sand. So another point that I am really interested in, that I've mentioned a couple times, I love talking about food. I love historic food. So that was something else that I was really interested in when I started this phytolith analysis. So I wanted to be able to talk about agriculture. So another pattern that's been kind of interesting is that I haven't found any identification of wheat, which wheat was, took a couple years for colonists to start growing wheat in Massachusetts. It also doesn't grow very well in Massachusetts. But I still thought it was interesting that I haven't found any wheat. And instead, I have found highly probable maize and bean and squash phytoliths. So of course, 3 native plants that were grown by the Wampanoag. But we're more and more kind of identifying as being part of the colonial diet as well. But it is really interesting to see these traces of maize, beans, and squash. And not really any typically colonial plants. So it's possible I will find some. It also is possible it could be a preservation issue. But that being said, it is still a strong pattern that I think is worth noting. So, and here on the bottom is, these are pictures of squash phytoliths. So they are quite distinctive. They have this like kind of scalloped appearance to them. They sort of look like tiny little cherry pits almost, but they're quite cute and quite distinctive. Okay, so if you're interested in phytoliths, there are a few really good kind of introductory guides to how to identify them, how to extract them. And another great piece that has really helped shape the way that I think about traditional ecological knowledge is a piece by Robin Wall Kimmerer. She's the author of Braiding Sweetgrass, which I highly recommend. It's a wonderful book. And also kind of what sparked my interest in this time and place and kind of questions is Changes in Land by William Cronin. So if you're interested in these kinds of colonial era climate and environmental change, I highly recommend that book. All right, thank you so much for the Peabody Museum and Mass Archaeology and Suanna for organizing all of this. And this is my email, anyagruber@utexas.edu. If you have more questions that you think of after the talk, I love it when people reach out with questions, so please do not hesitate to do that.
Thank you, Anya, for just a lovely talk, so informative. I think you hit so many different notes for people who are professionals and familiar, but for anyone in our audience today who is new to phytoliths and this aspect of environmental archaeology, a really wonderful summary of the contributions that this particular kind of deep and close look can offer an anthropological view of our past. We do have a couple of questions if you feel up for one or two questions today, right now.
Yes,
perfect.
Definitely.
So Louise wrote in just at the very beginning of your talk, and it made me think that thank you for introducing us to Rosalind. I think that's, you know, I name my tools. I think that's a wonderful tradition to develop that kind of a practice. But she's asking how, aside from maybe the microscopic perspective, how do you discern something as subtle as phytoliths inside of a soil sample?
So that's an excellent question. So when I am in an archaeological excavation, the way that I decide what context to take samples from and which context not to take samples from, I usually just go by the artifacts and the kind of soil patterns that we are seeing kind of broadly in that excavation. So what I mean by that is if we find a cellar, then I'll take samples from that cellar. If we find a house floor, I'll take samples from that. And I tend to just kind of over sample. So I will typically, sometimes it's a little bit excessive. I will generally just take samples from any context, any soil layer in an excavation that we deem important in some way. And sometimes that pays off and sometimes it doesn't. There have been many soil samples that I have extracted and gone through the whole lab process and done all the many steps it takes to actually extract the pollen, extract the phytoliths, although this happens with pollen too. And there's just nothing in it. So it really kind of depends, where sometimes the preservation there just aren't any phytoliths and sometimes there are. So hopefully that answered the question is I just take a bunch of samples and then I do all the stuff in the lab, and sometimes there's stuff and sometimes there isn't. And it's all just kind of partially just luck. And sometimes I mess it up and there are no samples, there are no phytoliths.
Very similar in geoarchaeology. We take
Yes.
more than we possibly can need. We hope for the best. And somewhere in between, we get some data to discuss. Thank you for that. In fact, Louise writes in a very big thank you. Love what you do is what she says. So second question is from Robert, actually two parts. The first part is, how do you distinguish naturally in place phytoliths from those that humans may have deposited? And are silicious diatoms similar to phytoliths? Could you maybe explain a little bit about the difference between those two?
Yes, so diatoms are similar to phytoliths, and I sometimes get diatoms in my samples, and I will note them, and I will say there are diatoms here. I don't do anything beyond that, just because it takes a lot of extra training that I have not done to actually identify the diatoms and figure out why they're important. So diatoms are definitely in a lot of my samples, but I just kind of acknowledge them and move on. But there are people who study silicious diatoms and can do a lot of cool work with them. I just don't know. I don't know much beyond knowing what they look like. So that's a great question. Also, how do I distinguish naturally in place phytoliths from like anthropogenically deposited ones? So that's another really good question. I take a lot of my cues and a lot of my interpretation from kind of the broader archaeological patterns. So if we're looking at a stratigraphic layer, that, for example, is like, I'm trying to think of an example, like a floodplain or something like that, then that is really helpful to me because the phytoliths alone don't necessarily tell me about the formation processes. It's really kind of the opposite, where the formation processes will help me understand the phytoliths. So I am very heavily dependent on interpretations in the field and our profiles that we draw in the field and our kind of post-field work interpretations and artifacts are very, very helpful to me. So I kind of have to have a very holistic view in order to understand those kinds of dynamics of what's naturally in place versus what was impacted by humans.
And you make a great case for that cross collaboration between specialists of different kinds inside of projects like the one that you're doing. That cross pollination, pardon the pun, is something that definitely helps. Thank you for that. Elizabeth asks the question: How have you come across any phytoliths from the past that are not found in today's Massachusetts plant communities?
That is such a fun question. As far as I know, I have not. What kind of distinguishes my work from many other paleobotanists is a lot of paleobotanists, people who study phytoliths or like leaf imprints or all kinds of other kind of botanical pursuits, is oftentimes they're working like from a million years ago or like many millions of years ago, and they'll find plants that are since extinct or that hasn't been identified in the modern day, or all those kinds of really cool stuff. And since I'm working in the scheme of things only 400 years ago, which of course is a long time, but in the schemes of a lot of the kind of plant populations that I'm looking at, I have not found any phytoliths. I'm trying to think. For pollen, I believe I found chestnut pollen, and we don't really have chestnuts. We don't have chestnuts really in New England anymore. I'm trying to think if I've ever found an elm pollen. Probably seen elm too. We don't really have elm so much anymore. But in terms of phytoliths, everything has been pretty recognizable. But trying to think. I don't think I found any phytoliths. Yeah, just those few pollen, which those trees, of course, still exist, but not so much in this area.
Well, I think that also sort of speaks to the really great resolution that archaeologists and specialists inside of environmental archaeology in particular, the kind of resolution that as scientists we can give discussions of the changeover of plant communities, and that speaks to climate and larger scale environmental change. So we get that at a resolution of almost a generational scale, where our colleagues who are doing ice cores and other things are looking at millennial scales. And so our resolution is certainly great fodder for interesting conversation and adding those humans back.
Mhm.
Let's see. I think those are all of the questions that we have for now. So I think we will conclude our program with great thanks to Anya for her presentation. Certainly a wonderful deep dive into Plymouth, Patuxet, and the histories that have been on so much on our minds with the commemorations of the 400th anniversary, and with surfacing and elevating Indigenous knowledge and Indigenous communities perspective on these histories. We do want to remind everybody that we will be back in 2 weeks. September 28th, we are joined by Katerina Menitti, who will be speaking on the distribution and use of Egyptian and Egyptian-inspired objects in Sicily during the Archaic period. Let's see, about 776 to 480 BCE, before the Common Era. So again, we want to thank everyone who's joined us this afternoon, and especially our speaker, Anya. We hope that we will be able to invite you back for future updates on your research, and to be able to cheer for you as you go through the process of completing the dissertation. And until then, please do consider becoming a member of the Mass Archaeological Society and joining us again for a future program of Diggin In, or through our other lecture series. So thank you again, Anya. Thank you again to all who joined us today.
Should I stick around for a second?
Sure thing. See if there's any last minute questions, burning scientific inquiry that has to come through today. Very good. I'm sure that you'll be able to update us in the future. There are a few more questions that we didn't get to today, but certainly I hope those inspire your work, and it will definitely inspire us to have you back to be able to talk about these in the future.
Yep,
So.
I did that.
Thank you, Anya. Take care.
All right, thank you so much.